All right, welcome everyone to the post-lunch session. It's my pleasure to introduce our speaker, Professor Sivaloganathan, who's coming to us from University of Waterloo, Canada. He is in the Department of Applied Math and also is the Director for the Center for Mathematical Medicine at Fields Institute in Toronto. Shiv has been visiting Bangalore. He's for IISc for 3 months. He's a visiting professor there and has started a new course on mathematical oncology at IISc, which he has taught earlier as well. Shiv has worked on diverse areas in mathematical biology, starting from hydrocephalus modeling and then getting into tumor modeling, thinking about drug resistance, tumor progression, all different kinds of multi-scale modeling approaches there, and has led multiple workshops as well, international workshops in this area at Fields Institute, including a program that is coming up this year, starting July to December. There's a 6-month program at Fields Institute, which Shiv will tell you more about. He's also an elected fellow of Society for Mathematical Biology and has received many other distinguished honors. Without any further ado, Shiv.
Okay, thanks. Thanks very much to IISc and to Mohit in particular for facilitating my visit, which has been great. It's great to escape Southern Ontario winters, January, February, March. I think I just go back for spring in Canada, hopefully. So what I'm going to talk about here is really something that the neurosurgeon I collaborate with has become very interested in over the past decade or so. And it looks like a therapeutic modality that may well become one of the gold standards in cancer treatments. At the moment, it's just chemo, radiotherapy, and surgery. But I think this has the potential to make a real impact in the field. So just before lunch or so, I was sitting with Jibin and Helen, and I wasn't going to put the next slide on, but Helen said, "No, I want to see the animation that Jibin made for you when he was a grad student." So this I used to put on and repeatedly used over the years as an illustration of the divide between the biomedical sciences and the mathematical sciences. I think certainly when Helen and I started in the field, the divide was quite clear. Let me just start also by just quoting Giancarlo Rota, who many of you may know. He was actually an applied mathematician who made seminal contributions in combinatorial mathematics and put that on a strong footing. And so I think years ago, it probably was true what he said, the lack of real contact between mathematics and biology. Is it a tragedy, a scandal, or is it a challenge? And I think now we can say that it is a challenge, and there are many of us working in it, and many problems that require a lot of joint efforts and thinking. So what I was saying initially, and this is, I have to thank Jibin for this animation, in the context of Canadian history, you could say that they were the two, what's the quotation? The two, I've forgotten what the quotation is, but two islands that never the two shall meet, and of course, two solitudes. That's right, the two solitudes where never the two shall meet. And so, when we started off, Helen and and I think I don't see any other senior people. You were probably not even born then. To work in this field was sort of a bit dangerous, because. Neither was mathematics nor medicine or biology. And so, when you applied for grants and things like that, you quite often get the response that, oh, this isn't mathematics and it isn't biology either. So at that stage, failure meant, that's it, you're finished. But the last decade certainly, and since Jibin's been a grad student, it's become really a two-way street. And I think it's now definitely true and standard to see teams of physicians, mathematicians, physicists, engineers, multidisciplinary teams working on problems. And there's been significant progress. And I think you could well say that this area, the applications of mathematics to medicine, has firmly taken center stage as one of the central areas of modern applied mathematics. Would you agree, Helen? Yeah? Yeah. So if I start by looking at statistics from 2017, You can see that 200,000 people or so would be diagnosed with cancer. And then if you look around here, it says 80,000 people roughly in 2017 would die of cancer. 1 in 4 Canadians will die from cancer. Percentage of people with cancer who are alive, 60%. And then there are 800,000 roughly Canadians who were alive at the beginning of 2009 with a cancer diagnosed in the previous 10 years. And then this frightening statistic, 1 in 2 Canadians will develop cancer in their lifetime, not necessarily die. But so it's a bit of a sobering thought to think that those statistics, even after 6, 7 years now, really haven't moved very much. So when you compare that with things like heart modeling, etc., at one point heart disease was the most sort of prevalent thing, and people died of it. But because I guess that problem is much more mechanical in nature, the advances made in heart disease, etc., and the modeling effects have actually had a much more dramatic impact. So I think now it's clear that cancer is the leading cause of premature death. It's overtaken heart disease. So there's a lot of, it's a sobering thought, and there's a lot of reason why you should get into this field, because this synergy between the mathematical and biomedical sciences is without doubt going to lead to, and already has led to, dramatic advances in clinical medicine. And you've heard this cliche before, but it will be similar to mathematics and physics at the turn of the 20th century, where that synergy and the interaction really led to huge advances. And I think that's what, so all of you who are getting into this field, you're just getting in at the right time, because we were talking over lunchtime about the way the field is at the moment is, in a sense, you could think of it as a pre-Newtonian sort of approach to medicine, because still the fundamental principles of growth and form, etc., in the biomedical sciences, nobody's, we may need the next Einstein to come up with it. But in the meantime, you can do this sort of thing that all of us do, which is to take experimental data, clinical data, etc., build a model where the parameters are fixed using that data. And then you take that model and go to a different experiment and use it to predict that. And now when that's completely wrong, you go, throwing the baby out with the bathwater. So you have to go back and think of it. And the analogy I've used all the time is it's very much like a portrait painter. If, for example, I was painting your portrait of you, it would be really broad strokes, right? The key features, and it wouldn't be every single detail. And if you do it well, then people will say, oh yeah, you've done a great job. And in a sense, this field is a bit like that. We sort of think by analogy, we sort of also use our experience, and then we hope we're picking up the key mechanisms driving the thing. And I know when I was a student, and certainly, the thing John Ockenden used to always go about, Occam's razor. So, really, the simplest model that can explain the situation is what we're looking for. And when you find that it doesn't actually capture the phenomena, then you sort of go back and see what you've missed. So it's a sort of gradual refinement. So the disease has been known since time immemorial. If you look at some of the historical records, you can find 3,500 years ago, there were documentation by medical practitioners of the struggle with the disease. And certainly the ancient texts from Greece and Rome show that early physicians were fully aware of cancer and capable of making a correct diagnosis. And Aetius of Amida, who was a physician in the Byzantine Empire, is an interesting guy, because he actually has 16-volume medical encyclopedia. And in one of them, there's even a prescription of how you do breast cancer operations. And quite clear, the knife should cut healthy tissue around a tumor, and a cauterizing iron staunch the blood. So some sort of awareness that it's not just the tumor. You want to make sure that cells haven't diffused or infiltrated the normal tissue. So what are the challenges in cancer treatment? You know, one of the things a number of people have talked about is the immune system evasion. You know, somehow they trigger various things, the cancer cells trigger various things that allow them to evade the immune system. And so immunotherapy, in a sense here, is trying to find ways of activating an individual patient's immune system to recognize and fight the cancer. Then, you know, people have talked about tumor heterogeneity. And cellular heterogeneity is really a crucial factor that complicates a lot of the cancer treatment response and, you know, drug resistance or drug-induced drug resistance. The tumor microenvironment, again, you know, you want to design a treatment strategy that's based on an individual patient's tumor microenvironment. And then drug resistance, there is inherent drug resistance, but there's also drug-induced drug resistance, and ways of overcoming this and modifying conventional treatments is one of the things. And then drug delivery. At lunchtime, we were talking to somebody about elevated interstitial fluid pressure and low blood perfusion, and these are all barriers for drug delivery. So how do you sort of overcome these? And there are a lot of questions. You know, some of the key questions are questions about the most efficient combinatorial treatment. How do you combine chemo, radiotherapy, or chemosurgery in the most efficient fashion? How do you overcome resistance to the immune system? So how do you modify the immunotherapy? How do you deal with genetic heterogeneity and what approaches? And then the drug resistance question and tumor penetration. These are all things that I think many of you and we for sure have been looking at for a while. But I want to focus on this particular new modality, which is high-intensity focus ultrasound. You know, I'm going to touch, I'll start with ablation, which is the easiest thing. But so here, the high-intensity focus ultrasound waves, actually, they could be, you know, anywhere between 10 and 500 watts per square centimeter. So what happens is here, the energy deposited by these high-intensity focus ultrasounds are sufficient to coagulate tissues. So if you're looking at a tumor, I'll show you how you know, how you actually approach this. Then you can actually essentially burn the tumor out. Then there's this interesting phenomenon that arises at certain range where you induce bubbles to form within blood vessels or other regions. And there's interesting potential applications of this cavitation phenomena. I put hypothermia here because it's actually talking to someone here at the Indian Institute for Science. Not necessarily even hypothermia, but sort of low intensity focus ultrasound. Somebody in mechanical engineering, Dr. Trivedi, who was telling me that their experiments show that low intensity will produce mechanical stresses on a cell. And if it's a normal cell, then the normal cell can withstand this thing and is unaffected by it. But they find about 60 to 70% of cancer cells actually go through apoptosis. They can't withstand the stresses. So this is an interesting, I never heard about this, and this would be something worth investigating mathematically as well. And then a colleague at Waterloo, who's an experimentalist, unfortunately I couldn't get the fast videos of it, but this phenomenon of sonoporation, where you use a bubble to sort of, you induce the bubble to break, releasing a jet stream, and then that jet actually perforates the cell, so you get a hole formed in the cell. So this is a way that you could deliver chemotherapeutic agents. And the interesting thing about this phenomenon is that you form the hole, and then within a short period, you know, nanoseconds, microseconds, or I've forgotten exactly the time scale, he actually has videos showing that the cell self-repairs. So as a way of delivering drugs, especially when you think about brain tumors, this is because one of the difficulties you have in delivering drugs to to treat brain tumors is that the blood-brain barrier is so tight that only oxygen and small molecular things can get across that. And so this sonoporation may be a way of delivering that. And so we'll just look at all those. And so let me start with some sort of background on this, focus ultrasound principles. So when you use ultrasound, you usually use this sort of focusing mirror or transducer. And I think you could probably trace this idea back to Paul Langevin as a physicist in the 40s. And he was the one who actually suggested this idea of a focusing mechanism for these high intensity things. And then Francis J. Fry and William J. Fry in the 50s actually experimentally demonstrated that this was feasible, etc. So the ultrasound itself generates two types of waves in tissue, and essentially longitudinal waves through the soft tissue. But when it hits bone, then you'll get shear waves generated along the bone. And the pressure is positive during wave compression and negative during rarefaction. So as the waves traverse tissue, you of course get attenuation, absorption, and scattering of the ultrasound wave, which deposits mechanical or thermal energy in the tissue. And as I've said, the waves are focused geometrically or mechanically or electronically to aim the energy emitted from the transducer to a very small target. And the acoustic intensity, the power focused over a small area, determines the amount of thermal energy absorbed at the focus. Okay, so essentially, if you look at ultrasound, I'm going to look at this simple direction where you're going to focus the ultrasound. You get vibration of molecules, energy absorption, temperature elevation, and then you get the tissue coagulation. I will also talk briefly, we haven't done a lot of work on this, but it's certainly a direction we're going on, this high amplitude threshold phenomenon of cavitation and their other related effects. So the high intensity focus ultrasound has numerous potential therapeutic applications. The one I'm going to spend a bit of time talking about is the ablative therapy. Then the blood-brain barrier disruption. You can think of this as a stable cavitation. So if you have a certain intensity of the ultrasound, and we're thinking about time harmonic pulsing, then what happens is the bubble starts expanding, it doesn't burst, and then it starts contracting. And the next, so this would be called stable cavitation. And you could sort of imagine that the stable cavitation is something that could pull the tight junctions of the blood-brain barrier apart a bit. And then this is the phenomenon that in sonoporation occurs, where you get unstable cavitation, where the bubble bursts and you get this microjet forming. And this, apart from sonoporation, is also, according to the neurosurgeon, even a potential application in clot lysis. So, when you have a clot, this would be something that could break it up. Of course, there's a lot more work required, because in breaking it up, you form lots of tiny clots, and this is something that could cause a stroke. But, you can see potentially that this therapeutic sort of modality could be very effective. Then, nanoparticle delivery. So I'll talk briefly about this. This is just work a master's student of mine recently completed. I won't go into a lot of detail, because it's just more deriving the equations and simulations. And genetic material delivery, and then this targeted delivery therapy, where you send nanoparticles in, and when they reach the location, you hit them with ultrasound to release. So this was an area, not with necessarily ultrasound, we hadn't thought about that then, but about a decade back, we did some modeling, and I'll say a little bit about that later. And then regenerative medicine, apparently HIFU, not at the highest intensity, apart from treating things like Parkinson's, etc. It also has some regenerative effect if it's not too high intensity. So those are directions we haven't looked at, but certainly potential future. Yep. No. So the stable cavitation, the bubble would expand and then contract. So you imagine if it's near the tight junction, it may compromise the tight junction a bit, but then the tight junction comes back, right, when the bubble contracts. So we were thinking, it's not anything we've explored, but we were thinking that's a potential way. But maybe sonoporation may be more effective, just puncturing a hole through the cells, delivering the drug, and then the cells close by themselves. So there are, I think at the Riken Institute in Japan, there's a group led by Shu Takagi, who actually do full three-dimensional computational simulations of this high-intensity focused ultrasound. So this is, you can imagine when you do the high-intensity focused ultrasound, the skull will also refract the rays. So you have to have some computational way of actually focusing properly within the brain. So this is a setup that, my long-term collaborator, James Drake, who's the chief neurosurgeon at the Hospital for Sick Children in Toronto, this is the experimental setup he has. So you have this magnetic resonance machine here with a patient thing. And then I'll show you another diagram which shows where the transducer is placed. Okay, it's in the patient table. And then you have this high-intensity focused ultrasound planning console, which allows you to focus the ultrasound beam precisely. Because you can imagine if you have a tumor, the focal point is sort of smaller than a grain of rice. So you want to adjust it to sort of ablate the whole tumor. And then there's a magnetic resonance console. Magnetic resonance imaging, you all know, but you can actually adapt it to do magnetic resonance thermometry. So essentially what happens here is you have, it uses the phenomenon of proton resonance frequency shift, if there's water content, and you can accurately map the temperature within soft tissue. So around the tumor, you can make sure that you're not elevating the temperature in healthy tissue too much. Now, the experiment that Jim's doing is actually on bone sarcomas. So this is directed at bone sarcomas. But I just wanted to say that, if you look to the 70s, William Fry and Carl Townsend were people who actually pushed this field a bit forward. And so I think round about the 80s, focused HIFU gained a bit of traction. And it sort of really took off in the 90s when the FDA in the States actually approved it for prostate tissue ablation. And now in the 21st century, it's sort of going full steam ahead because now you can couple all of this with robotics so that you get extremely precise things. And one of the attractive features of this is that it's a non-invasive thing, right? You don't have to go in and surgically remove, etc. And hopefully at the end, I'll show you a video of about 3-4 years ago when Jim did the first HIFU operation on a bone sarcoma on a young kid. And it's quite remarkable. So this is when we started getting involved. And I'll show you. Okay, so this is the patient bed there, and that underneath that, there's a hole there, is this transducer's place. And the transducer is controlled here, and you can change the angle and focus it quite precisely. And then temperature mapping, etc., through the MR thermometry. So as I said earlier, it's a very tightly focused ultrasound. You're looking at 10,000 watts per square centimeter. And when you compare that with medical imaging, the low intensity, that's 0.001 watts per square centimeter for diagnostic ultrasound. So this really has significant heating and mechanical effects. There are all sorts of things experimentally you have to do to make sure that you don't generate shear waves at the skin interface. You have to put water, etc., so the transition is gradual. Then the challenges of bone MR thermometry HIFU is that okay, I already mentioned this, the fact that that when you get to the bone, there's very little water content in the bone. And so one of the worries of the neurosurgeon is, okay, you burn that tumor on the bone surface. What happens, how does the heat dissipate within the bone? And does it affect the bone marrow and other important aspects? So that's something which you can mathematically model, especially when you see the data. You find that temperature, away from the bone a little way is actually normal body temperature. It's not elevated that much. So you could think of posing a simple problem where the cortical bone is a sort of circular cross-section. Take a Dirichlet temperature condition far from the bone, and right where the tumor is, you have this sort of delta function heat, and then solve the heat equation, except now you're looking at a porous medium, right? The bone. Bone is actually porous. So you want to extend the heat equation in some way by homogenization, and then you could predict the temperature distribution. So here's the simple model that we're looking at. And in the neurosurgical literature, they call it the Penn bioheat equation, but in fact, it's just the heat equation, right? It's, the normal form you see of the heat equation is that, the homogeneity and isotropy of the tissue. But here, I've left the diffusion tensor there, K, in the hope that you could use something like diffusion tensor imaging to get a more accurate representation of how the heat would diffuse. And this, you can think of it as something like Newton's law of cooling. You've got the blood temperature. If you elevate the temperature above something, then there'll be a sink term that removes heat from here. And this Q here is really the source term generated by the acoustic pressure. So you have acoustic pressure there. We'll be able to work out what the source heat term is there. And so if you look at the experimental data, then this is a purely experimental heuristic formula I'm going to show you here. And don't ask me where the 43 comes from, but in the medical literature, they all use 43 degrees Celsius. But it gives you the dose equation. In other words, suppose your high-intensity focused ultrasound was such that you're raising the temperature to 50, say to 44 degrees or 45 degrees, then the energy deposited there obviously is going to be more than if you were elevating the temperature to 43 degrees. So the thermal dose equation really gives you some equivalence. Okay, so how long should you pulse the high-intensity focus ultrasound to get the same result? And so, for example, it tells you that you need 120 minutes at 44 degrees, 60 minutes at 45 degrees Celsius. And if you're at 53, 1 second, something like that. So 43 appears to be an arbitrary choice, but above trend seems to be conserved across multiple cell types, though the heat sensitivity, of course, will differ. And the model seems to be valid for the high temperatures in high-intensity focus ultrasound. Of course, the threshold for thermal dose required for cell death varies for tissues with different thermal sensitivities. So that's the thermal dose equation. So I said 240 minutes at 43 causes thermal necrosis in soft tissues. 1 second at 57 produces thermal necrosis. So, of course, as a mathematician, you're sort of far from satisfied from just seeing some formula that's just been thrown there from bunches of experiments. And I think you can actually derive something like that or an alternative for the thermal damage equation using the same logic, but now using an Arrhenius type relation where the rate of chemical reactions related to the temperature. And so here the EA is the activation energy. I think, and there are modifications of it, I believe you can actually derive a thermal dose equation starting from here. and maybe even a similar thing to what the physicians use. Okay, lastly, I haven't put the wave equation, the linear wave equation. Okay, so the linear wave equation gives you the acoustic pressure, which goes into the heat equation as a source term. Now here I'm talking about harmonic pulsing. So if you take the wave equation and do separation of variables, where now it's t, capital T, a function of t, times the p, which is a function of the spatial variables. Then we say that it's harmonic pulsing, so d2 capital T by dt squared is equal to minus omega squared t. If you do that, then substitute it in, everything cancels out except for the spatial component, and that gives rise to the Helmholtz equation, okay? So essentially, we're doing time harmonic pulsing, and so you really just have to solve the acoustic pressure equation given here by the Helmholtz equation. And you can do it using Green's functions or whatever way. You can write down an expression for the acoustic pressure, okay? And so this essentially is what's being used in the experimental setup. Phillips has sort of programmed something like that in. And the bone microstructure, remember I was saying, it is a porous medium. So when we want to determine the heat distribution, when you elevate the temperature to a delta function of the boundary, then you really want something more than just the standard heat equation. You want the heat equation extended to a porous medium. So you can do that through homogenization. So don't worry about this. You know, if you're a mathematician, you're always thinking about mathematical generality and well-posedness. And so you can write that system of equations in a slightly different form given here. And you can, you know, using Darcy's law, etc. You get this equation. And then to prove well-posedness, you want to establish existence and uniqueness of a solution and continuous dependence on initial data. And so this is a formulation in theoretical PDEs. And then you can apply the so-called Array-Schauder principle. Yeah, don't worry about that. That's, so if you're interested in the mathematical details, this is in Advances in Mathematical Sciences and Applications, the proofs of that. So you can actually go even further. You can actually prove the existence of a uniform attractor of the model. And we did that and estimated the Hausdorff dimension in terms of the model parameters. And that's in the Bulletin of Mathematical Biology, which is surprising because usually I think we motivated it enough with the fact that this has a real application. But, so this is the thing that Helen, Gibbon, everybody went on about, and probably worth emphasizing in all of these areas is the major challenge is the multi-scale modeling in medicine and biology. Because we know from many of the talks that have come through that, you know, when you get a tumor forming at the organ level, you know, there are mechanical forces that are exerted. And there's mechanotransduction. So you get actually information that gets transferred all the way down to the gene level. And it can cause alterations at the gene level. Similarly, at the gene level, there are sort of certain genes activated for various reasons that will sort of have repercussions right up there. So there's a two-way flow of information. And the million-dollar question is how to couple information at every level. Now, as Gibbon said in his talk earlier, you know, sometimes the questions you ask maybe at this level or at that level. Then you want some way of being able to incorporate some of the information from above and below. And so in that sense, you want a framework where you can have modular inputs at each level. And depending on the question, you would focus on that. And it also emphasizes the same sort of thing that everybody has been saying, that you can't really have one team sorting this out. You know, you really need experts in various areas depending on what the question you want to ask is. But in the meantime, while we're trying to work out you know, how information flows here, there are lots of low-hanging fruit questions that you can ask and that you can answer. I think when I was talking at the Indian Institute of Science, I sort of explained why I put this up, and maybe I should tell you as well. you know, at Waterloo, one of the things that's happened there is that we have this Perimeter Institute, an Institute for Quantum Computation, and you know, a lot of bright students like you would all finish their undergrad and say, I want to do cosmology or string theory, you know, how the universe develops. And so this was my pitch that the challenges in inner space, okay, there, are just as challenging, if not more challenging, than outer space. And so I like to call this the grand unified theory, you know, and so it's partially worked because we've had some very good students who've decided they'd rather work on this than, you know, what happened in the first 10 to the minus 40 seconds of the universe. So here are the challenges, again, which Jibin emphasized again. It's challenges in the multi-cell modeling which are over length scales. You know, if you think of the person, you're thinking of order of a meter, electrical length scale of cardiac tissue, 1 millimeter, cardiac sarcomere, 1 micrometer, and then pore diameter of a membrane protein, 1 nanometer. So that's a range of 10 to the 9 in the spatial scales. And if you look at the time scales, it's equally daunting. There's 10 to the 15 range. So these are the challenges we have to face when we're thinking about modeling. In some sense, you can sort of, these are just pictorial things. You can read many papers that look at it. But, you know, one of the classical things, if you look, is propagation of acoustic waves through a bubbly fluid. A lot of work was done in this in the 40s and 50s. And you actually find that there is work that's given rise to sonar in oceans, you know, which works very well, the modeling. Although the scales are very different, the mathematical models for looking at tissues and bubbles formations are pretty similar. Okay, just the scales are different. Here, if you look at the oscillation of a single bubble, on the intermediate scale, you've got lots of bubbles and they're oscillating, right? And then on the coarse scale, what the bubbles actually do is make the medium act as an effectively compressible material medium. And then if you look at blood vessels, again, the fine scale corresponds to flow down a single vessel. On the intermediate scale, you've got the vessels acting like a discrete network. And then on the coarse scale, what happens is the vessels actually make the material act as an effective porous material. Okay, so you need mathematical tools for handling those things. And so what I was saying here is that the fine scale corresponds to wave propagation through a periodic array of bubbles, right? And then B, the bubbles here act as an effective compressible medium. If you're looking at the blood vessels, so maybe at this point I should say, if you look in material science, there's a lot of work that's been done in material science looking at the atomistic level, and then to dislocations and crystals, and then crack propagation, etc. And so they've done a lot of stuff, but I think the mechanics community there hasn't taken up the challenge in the biological or biomedical sciences. And these are major challenges, mainly because the individual units or the cells themselves are very dynamic. So no two cells will be exactly identical. You know, so you have to think of ways of, for example, when you're looking at the vasculature, we know that you know, the vasculature looks very different. So each of the little units here may not look identical. But what you can do is use things like the method of multiple scales and impose the condition that even though they look slightly different, they're periodic. Okay, and then you get this material that's sort of homogenized. And so that even though the details of the structure may vary from point to point, you can assume some sort of periodicity to fix that degree of freedom. Okay, so here I'm going to now talk a little bit about the nanoparticle, or in the context of imaging and drug delivery. And this is from, Wilson and Byrne's, so this is from Wilson and Byrne's 2010. you've got these sort of imaging techniques using ultrasound, which is sort of given these pictures. I can't actually identify, where the tumors are, but it's sort of pointed out there. And now, if you think about microbubbles in a medium, you can sort of blow it up to a macro bubble. But we're sort of going to try and think about the ways of imaging that have been produced. So this B mode imaging is a method of using ultrasound that actually shows up as bright spots. So you can actually get structures quite accurately using this B mode. And then the power Doppler approach is actually one where you can visualize blood flow down small vessels, etc. So different modalities of imaging. So unfortunately, I couldn't get the video from my colleague, which didn't seem to work. But the general concept here of sonoporation is that you have a bubble that is near a cell membrane, or say it's induced through cavitation, and then what you do is you it's unstable cavitation. So the actual bubble bursts, and the pressure jet then punctures a hole through the cell membrane. So here's a picture from Alfred Yu, my colleague, that shows here for a living cell, when you do these this sort of sonoporation, you get multiple holes there in the thing. And his live video actually shows one hole being punctured, and then over time you see it closing up again. Yeah, this is actually from the video where he's punctured a hole, and then that hole then re-repairs. And so this is some cell line, again, I don't know, MRC5 fibroblast, the diploid cell line. And you see there, I think you can see it sort of closing a bit here. So this is part of the video. And then, so you get localized membrane perforation and recovery. And so this is quite an amazing technique. So the event sequence is essentially a single pulse ultrasound. The microbubble ruptures, and then you get a local membrane pore, and then membrane reseals. So again, I've talked about the stable cavitation. The bubble pulsates over many cycles, but remains physically intact. And it gives rise to low amplitude acoustic responses. And then, of course, the inertial cavitation, here the bubble expands, collapses rapidly. It's also known as transient cavitation. And there's a release of various high amplitude acoustic responses from shock waves, sonoluminescence, etc. Interesting thing is that although the mathematics, not specifically for this, but looking at bubbles and the phenomena that arise, was developed by Brooke Benjamin and others in the 50s. So if you look in the Journal of Fluid Dynamics, you can find mathematical models that may be transported into this field. So the two acoustic parameters that directly contribute to cavitation dynamics are the peak rarefaction pressure. A large value favors gas cavity expansion. And also ultrasound frequency. The smaller the value, the longer the wavelength that you get. And the wavelength's much bigger than the bubble size. And interactions with the bubble then becomes more acoustically homogeneous if you take longer wavelengths. So this is just going to say a little bit in the last 10, 15 minutes about the problem that my student was working on, about acoustic droplet vaporization. We're really thinking here about a drug that's being taken to some location, and then you're using ultrasound to vaporize it and release whatever's there. So the therapeutic applications, as I've said here, are targeted drug delivery and the so-called embella therapy. And the diagnostic applications, you can use the bubbles. Because basically, when you pulse ultrasound through, the bubbles themselves will generate a response which is nonlinear. The actual ultrasound wave that you pulse in, when it meets tissue, etc., and bone, there's a reflected wave, but the responses are linear, essentially. So if you look at summing the ingoing and outgoing things, the stiffness of the material, etc., linear responses, they all cancel out. So that the bubbles actually enhance that area that you're sort of trying to image. So here's a thing from Rakesh Jain back in, I've forgotten what paper, 2001. So he had this idea, which I don't know everybody agreed with, but if this was a normal tissue and normal vasculature, what Rakesh Jain and his co-workers said is, the tumor, as we all know, generates very abnormal vasculature and sort of attracts, and basement membranes leaky, etc. And so that also makes administration of drugs a bit difficult because there's perfusion's not that great through a tumor vasculature. And what he said in this paper then was that, in healthy tissue, you've always got anti-angiogenic and pro-angiogenic factors, which are in balance. And so we're all healthy, thank God. And in sort of cancers, the pro-angiogenic factors that sort of outweigh the anti-angiogenic factors is assumption. And so if you were to administer anti-angiogenic factor, then he said this would sort of balance things out. And he said that there is a certain period of time which he called the normalization window, where all the vasculature in the tumor has been normalized. So in other words, this is when you should put the chemotherapy, administer the chemotherapy, because it will actually reach all the locations, and that would have the most impact. So I think probably before Jibin came, or around about that time, we were looking at a therapeutic thing that Shiladit Sengupta, who I think was IISc and now at Harvard, he developed these nanoparticles that inside were the cytotoxic drugs, and then pegylated on the outside of the nanoparticles were the anti-angiogenic therapy. And they both had different release kinetics. And experimentally, you see, so when Rakesh Jain said this, there were a lot of people who would administer anti-angiogenic therapy and then sequentially then wait and then administer in this normalization window the cytotoxic therapy. What Shiladit showed was that putting the nanoparticles in actually has a much more dramatic effect. So we, at that stage, tried modeling that. And so this is the nanoparticle delivery with the, so this was the dynamics of tumor growth and combination of anti-angiogenic and cytotoxic therapies using nanoparticles. And, it agreed with Shiladit's results. It is indeed better to do this than to sequentially administer anti-angiogenic than cytotoxic therapy. But you see, when you think about it, sometimes, mathematical models really tell you something that intuitively is sort of obvious, right? Because if you look at, for example, this scenario, suppose you sequentially administered first the anti-angiogenic therapy. Well, it would come down here and it would actually cut quite a few of these vessels here, right? That's what anti-angiogenic therapy sort of cuts the vasculature. It sort of stops sprouting, etc. So now, if you were to administer cytotoxic therapy, well, there are vessels that have been cut, in other words. And so, although you may have some effect, it may not reach all the locations. Whereas if you had the nanoparticle with the pegylated anti-angiogenic therapy and the cytotoxic therapy, well, that reaches as far as it can. It releases the anti-angiogenic therapy and cuts the vasculature, but the cytotoxic drug is already there on location. And so then it has the optimal effect. So that sort of heuristically explained why everything tied in. So the contrast-enhanced ultrasound. Conventional ultrasound encounters, difficulty, where is it? When imaging tissue vasculature. So this is where the contrast agents such as microbubbles can improve contrast in the resulting image. Now the microbubbles, however, lack stability in circulation. Liquid droplets have also been proposed. They have increased stability within the bloodstream, but they have a lower contrast enhancement effect. And then phase contrast agents combine the two using ultrasound to trigger the vaporization. And so this is really what my student was looking at. And what he decided to do was, if you're looking at ultrasound axial resolution, the axial resolution really depends on the spatial pulse length, i.e. number of cycles per pulse. And you can sort of convince yourself why that's the case, because then you get the reflected, that you can't distinguish between the reflected and the incident waves. And so what he did was to look at this chirp ultrasound. And so this is a way of increasing axial resolution without sacrificing total transmitted power and depth. And what this essentially does is what you see here. It varies the frequency, with time. So it's not a fixed frequency. And that allows, if you think about it, it sort of captures the lower frequency, which will have greater depth, but and the higher frequency. And actually, it doesn't come to mind immediately, but it also gets rid of some of the noise artifacts that arise in these cases. And so you want to mathematically set this up. So, we always like radial symmetry because it's sort of, so here's your bubble with, and it's just 1D, I love 1D problems because I think I can solve them, but anything more gets a bit difficult. So if you want to model acoustic droplet vaporization, you want to look at the evolution of the radius of the bubble over time. And you want to take into account the vapor behavior within the bubble, mass flux and heat transfer across the bubble surface, and then heat transfer across all three mediums, PFP liquid, shell, external water. So here we're looking at a shell and so you really want to also break the shell at some point, right? When you're trying to deliver something. So the fluid flow within the internal, external, and the elastic response to the shell. And so we won't spend any time here, but you start with the Navier-Stokes equations, and take spherical symmetry. You integrate Navier-Stokes from R to infinity. Apply conservation of mass, continuity of stress to get what's known as the Rayleigh-Plussett equation. And the Rayleigh-Plussett equation is really key in a lot of these studies to study bubble dynamics, sonoluminescence, all of that. You need to look at the Rayleigh-Plussett equation. And then the vapor pressure within the bubble, a student decided instead of using an ideal gas law to use the van der Waals equation. And evolution of bubble surface temperature, again, you can calculate. It's better to go and sit down with an hour or two to look at the equations a bit more carefully. But you can then look at the evolution of the temperature profile, which is given by this energy equation. And you have standard boundary conditions. And the shell he modeled as a hypoelastic solid. So again, that's all standard theory that you can look up, and the shell response, etc. So this is the constitutive relation for the hypoelastic solid, a Mooney-Rivlin strain energy density functional. And the shell, there's no point in going into all of this detail. I just want to show you some of his simulations that he did for this model. So don't ask me what the cash-carp method is with adaptive time steps, but it's some stable numerical method for solving these types of equations. And the energy equation governing the evolution of the temperature profile was solved using Crank-Nicholson method across the computational domain. And then he has certain terminating conditions. And so essentially, you're looking at pictures here where the pressure, ambient pressure is 1.5, 2.5, 3, and 4 megapascals. So I think essentially we want to avoid really having too many bounces, okay? Because you'd like the thing to just vaporize and go. So here, if you look at the linear chirp expression, this is it bounces once and then it sort of vaporizes. The 3 megahertz, 6 megahertz, 4.5 megahertz are the other curves. They also all bounce once. Now, here, so this would be, I think, is that 2.5? I'm not sure which the ambient pressure is, but you can see here that the linear chirp, which is the black line, actually goes through almost a bounce, then bounces and then vaporizes. The other ones seem to be doing a bit better, right? So these are just, I mean, we haven't experimentally done anything here. This is just his simulations, which he wrote in his thesis. And here's something I guess you really don't want, too many bounces, it's not really, so if you're thinking about drug delivery, you want it really to break and then the drug delivered, okay? So this was, I think that was my time to quieten down. So anyway, so those are all just simulations with no match to experimental work. So this is just my conclusion, saying that mathematical modeling, if you look at molecular biology, it is increasingly focused on complexity of pathways and targeted therapies. And sometimes you look at a pathway here, which actually, even if you stop that pathway, there are other pathways that, so you haven't really attacked the basic problem. So, my approach has always been from the other end. So you can use mathematics to develop multi-scale models, and even looking at the macroscopic level to simplify and present credible hypotheses for clinical trials, and to investigate novel interventions and therapies. So I just wanted to say to all of you, yeah, the future is so bright, you've got to wear shades. And I want to thank, of course, the National Science and Engineering Research Council in Canada, which has really funded all my research since I've been there. The Canadian Institutes for Health, these collaborative types of things in Canada are funded by Canadian Institutes of Health Research. Fields Institute, which is, paid for workshops and postdocs, etc. And Indian Institute of Science, the Axis Bank Center, and Mohit, who facilitated this visiting professorship. So these are just some, there's too many to mention, but these are people who are directly involved. Kevin Jiang is the one who worked on the last bit on chirp, ultrasound, etc. Karina, who's one of my early postdocs, and Jibin here, who sort of could have taken this hour as well because he looked like he hadn't enough slides for another 2 hours. And collaborators, which include Marin Carter. Marin Carter, you might know of the KPZ fame, the Carter, Parisi, Zhang. He helped in our modeling of the anti-angiogenic and chemotherapeutic. Jim Drake, who's the chief neurosurgeon, Amit Oza, who's the chief oncologist at Princess Margaret Hospital, Mike Milosevic, Richard Hill, and John Dick, who's really one of the pioneers in cancer stem cells and dedifferentiation, etc. And well, thank you. I think I'll stop there. Any questions? Although I've zoomed through a lot of stuff. I think if any of you are looking at interesting problems to look at in this interdisciplinary field, I'd suggest, definitely high-intensity focus ultrasound and cavitation or sonoporation. I think there's a lot of mathematics that was developed, not in this field, but just in fluid dynamics, that can be translated and used in a context here. So. Yes.
Drug delivery, right?
Yes, yes.
It's similar to...
Similar, so this modeling would apply to that as well. I mean, Kevin just looked at a shell because we were thinking of encapsulating some drug, and then you just break the shell.
Are they using lipid-based drug delivery?
Yes, in some cases, but it's nothing that's gained a lot of traction. I think there's sort of instability of those, and so if you could find something more sort of stable and. I think they all need a cup of tea or coffee to look after lunch. Sort of. Oh. Okay. Oh, so I needed to cut it short, but anyway, that's fine. Oh, no, that's fine. No, there's just other things I could talk about, but that's fine.
Okay, so then maybe we'll break for a little bit. And then